Hi. Today we're going to discuss the basic principles of heredity. These include the laws of allele segregation that were established by Mendel. We're also going to discuss some probability and how it relates to genetics, and also some statistics. So I hope that you will find this interesting and think about what I'm saying in the lecture and prepare some questions for question time. And now, here comes the voiceover. Today, we're going to talk about the basic principles of heredity, and we're going to be focusing on Mendelian genetics. Here is the garden pea that was so useful to Mendel. Mendel lived from 1822 to 1884. He observed the inheritance of a few select traits of the garden pea. Using his observations, he proposed the basic principles of heredity in 1866. He is known throughout the world as the founder of genetics. His work was unappreciated for 35 years until it was rediscovered by Hugo de Vries, Karl Correns, and Erich von Tischermach in 1900. With the rediscovery of Mendel's work, others made the connection between heredity and disease. One of the most famous people to do that is Sir Archibald Garrod, who lived from 1857 to 1936. He worked on Alkaptonuria and other diseases. He observed that these diseases appeared at birth and remained for life, that the parents were frequently first cousins. So he proposed in 1902 that this disease was a rare recessive gene disorder. He was right. He proposed that each gene codes for an enzyme, and that diseases were caused by inborn errors of metabolism. His ideas were ignored for 30 years, and of course, his work did not exist in a vacuum. He had access to Mendel's ideas, which were rediscovered around the turn of the century. So, what is alkaptonuria? It is an inherited condition that causes urine to turn black when exposed to air. It results in blue-black pigmentation occurring in the eyes, cartilage, and skin of those affected. People with the disease typically develop arthritis, particularly in the spine and large joints, beginning in early adulthood. It's a rare autosomal recessive disease with 1 in 250,000 people affected approximately worldwide. Although alkaptonuria is more common in certain areas of Slovakia, where it has an incidence of about 1 in 19,000 people, and also in the Dominican Republic, where it has a higher incidence. The disease is caused by mutations in HGD, homogentisate oxidase, an enzyme that helps break down the amino acids phenylalanine and tyrosine. So when did genetics really become a discipline? William Bateson, who lived from 1861 to 1926, worked with poultry and sweet pea. He is the chief popularizer of the ideas of Gregor Mendel following their rediscovery in 1900 by Hugo de Vries and others. He coined the terms allelomorphs, which was later shortened to allele, heterozygous, homozygous, and in 1905 he suggested the new word for the study of inheritance, genetics. He wrote, "Such a word is badly wanted, and if it were desirable to coin one, genetics might do." This brings us to some important genetic terms. And these are some terms that I would like you to know as we proceed through today's lecture and also through the rest of the class. Trait, an inherited characteristic. Gene, a genetic factor, region of DNA, that helps determine a trait. Allele, one of two or more alternate forms of a gene. Locus, specific place on a chromosome occupied by an allele. Loci, plural of locus. Genotype, set of alleles that an individual possesses. Heterozygote, an individual possessing two different alleles at a given locus. Homozygote, an individual possessing two of the same alleles at a given locus. Phenotype, the appearance or manifestation of a trait. Epistasis, the genetic interaction of two independent traits. So why was Mendel so successful? One of the main reasons was his choice of model organism. Here, pictured on the left, the garden pea has one growing season, it's an annual. It's considered fast. The plants produce a large number of offspring. And he was wise in what characteristics he chose to follow, what traits he looked at. He looked at 7 easy-to-see phenotypes, and there were no epistatic interactions between these genes. He also pursued a hypothesis-driven experimental approach, which was very helpful for him to understand his results. And he patiently conducted and recorded for 10 years before publishing. So there was no pressure to publish, no rush to publish. He studied, he observed, he planned, he worked, and then he formulated his ideas and published them. So what are the traits Mendel followed? Seed color, either yellow or green. Seed shape, either round or wrinkled. Seed coat color, either gray or white. Pod color, either yellow or green. Pod shape, either inflated or constricted. Flower position, either axial, which is along the stem, or terminal at the tip of the stem, or stem length, either short or tall. Using these traits, Mendel was able to formulate his principle of segregation. Each individual organism possesses 2 alleles encoding a trait. Alleles separate when gametes are formed. Alleles separate in equal proportions. So how does this work in a diploid organism? We're going to start to get into this now. Let's look at one of the traits that Mendel looked at, seed shape. Genes can exist as different versions called alleles. And for seed shape, there are 2 alleles. The round allele, which is denoted by an uppercase R, and the wrinkled allele, which is marked by a lowercase r. These alleles occupy the same position in a chromosome because they are alleles of the same gene. You can see the big R is on the left-hand pair of this pair of homologous chromosomes, and the little r is on the right-hand pair. The position of the alleles is called the locus. Diploid organisms can be either heterozygous or homozygous for a given set of alleles. The pair of chromosomes here on the left has 2 different alleles of the seed-shaped gene, the round allele and the wrinkled allele, and is therefore heterozygous. The one on the right has only one type of allele. They're both round. So this one is homozygous. The genotype is related to phenotype, but some alleles have a more pronounced effect than others. Here we have 2 homozygous pea plants for the 2 seed-shape alleles, round and wrinkled. From this, we cannot tell the relationship between these 2 alleles. But what if we cross them? By crossing purebred round and wrinkled pea plants, we can determine if an allele is dominant or recessive. By crossing a parental generation or P generation, where one parent is homozygous for the round allele and the other parent is homozygous for the wrinkled allele, we look in the next generation, which is called F1, we get all phenotypically round seeds. So that means that the heterozygous animal here has a round phenotype. So round is dominant over wrinkled. Wrinkled is therefore recessive. Recessive alleles are traits that are not expressed. So if we do a monohybrid cross with these same parents, round and wrinkled, the F1 generation, or first filial generation, will be all round. So our first observation is that wrinkled, that phenotype, was lost in the F1 generation, and that round is dominant, and the character that prevails is dominant. Now, if we cross these to each other in the F1 generation, so we self these, which is something you can do in pea plants. In the F2 generation, you will get both round and wrinkled seeds. So how can this be explained? Since wrinkled was seen in F2, F1 must carry the recessive allele. Mendel actually carried out these crosses, and he found that he got 5,474 round progeny or offspring and 1,850 wrinkled progeny. And that gives us the famous 3-to-1 ratio. The 3-to-1 ratio was predicted by Mendel's theories. Let's look at this cross again. We cross the homozygous, either round or wrinkled parents. In the F1 generation, we allow the plants to self, and all the F1 generation is round. If you allow the F1s to self-fertilize, as before, you develop two types of gametes, either round, big R, or wrinkled, little r. In the F2 generation, the first gamete is in purple, and the second gamete is in black. This yields 4 genotypes: round round, round wrinkled, wrinkled round, and wrinkled wrinkled. This gives a phenotypic ratio of 3 round and 1 wrinkled. And this illustrates Mendel's first law, that the two alleles separate at equal probability. So let's do a test cross. You're given a round seed and asked to figure out its genotype. Round seeds can have two potential genotypes: round round, both round alleles, or round wrinkled, a round allele and a wrinkled allele. We cross this genotypically unknown seed after it's been planted and grown up into an adult plant with a recessive parent. So that parent, we know its genotype, it is wrinkled wrinkled. If the genotype of the seed was round round, you should see that the progeny from this test cross is all round. If the seed was heterozygous, you should see the ratio of the progeny should be 1-to-1, 1 round and 1 wrinkled. A cross between an F1 genotype and either of the parental genotypes is a backcross. Mendel tested his theory of inheritance of dominant traits by first crossing homozygous round and wrinkled parents. This generated heterozygous F1 progeny, as before. Then, crossing the F1 parent with either parent, you got different results. If you cross an F1 with P1, which is homozygous round, you get all round progeny. If you cross an F1 with P2, which is homozygous wrinkled, you have a 50% chance of getting either round or wrinkled offspring. This was further evidence that supported Mendel's theory of inheritance of dominant traits. The Punnett square is an easy way to see how alleles segregate. It was developed by Reginald C. Punnett in 1917, and I imagine you've probably come across this before in your scientific career. So if you have a P generation where you have homozygous round and homozygous wrinkled parents, and then you allow the next generation to self, in the F2 generation, you get different possibilities of gametes. So you can put them on the Punnett square, which here, since there is 2 alleles, you have 4 different positions for those alleles to go into. So you make a Punnett square like this. You put the possible alleles from the first gamete on the left and the possible alleles from the second gamete on the top. Then you add these gametes together. So you get round, 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 wrinkled, wrinkled, round, and wrinkled, wrinkled. By simply counting, we can determine the types of progeny produced and their ratios. So we get 1/4 are round, round, 1/2 are round, wrinkled, and 1/4 are wrinkled, wrinkled. So genotypically, we have a 1 to 2 to 1 ratio. Phenotypically, of course, we have a 3 to 1 ratio. 3 round-seeded plants to 1 wrinkled-seeded plant. Probability lies at the basis of what we have been discussing. It is useful for you to understand a little about how probability works. Here we have the most famous tool for games of chance, the dice. The law of probability that governs outcomes with dice also governs outcomes in genetics. Probability P is the chance that an event will occur. Probability equals the case A divided by the total number of cases N, or P equals A divided by N. This assumes that all cases are equally likely events. One of the rules of probability is the multiplication rule. The rule states that the probability of 2 or more independent events occurring together is calculated by multiplying their independent probabilities. Here's an example. There are 6 sides on dice. Let's say you roll a 4, which is depicted on the right. There is a 1 in 6 probability, 16%, of getting a 4. Roll again, and the probability is the same, 1 out of 6, 16%. But getting 2 subsequent rolls of 4 is more difficult. The probability is multiplied, 1 over 6 times 1 over 6, which equals 1 over 36, or 2.6%. So you can see it is much less likely to get 2 of the same events happening one after the other. The addition rule states that the probability of 2 or more mutually exclusive events occurring is calculated by adding their independent probabilities. I turn to dice again for another example of how the addition rule works. You add the probability of each event. So let's say you want to get a 3 or a 4. The probability of getting a 3 or a 4 is 1 over 6 plus 1 over 6, which equals 2 over 6, which equals 1/3. So if you're willing to accept 2 different kinds of outcomes, you're more likely to get what you want. Let's explore the law of probability using an example from Mendel. Here we have the segregation of stem length, and there are 2 different alleles. The short allele, which is a lowercase t, and the tall allele, which is an uppercase T. Let's look at the segregation of these alleles in terms of the law of probability. So if we cross 2 pea plants that are heterozygous for the stem length alleles, we will get 4 types of gametes, 2 from one parent and 2 from the other parent. And these will occur at equal probabilities. These gametes can then produce offspring of 3 genotypes, with the heterozygote being the most common. So what are the chances the offspring will be short? 1/2 times 1/2 equals 1/4 of the time. That's a 25% chance. What are the chances the offspring is heterozygous for the height gene? 1/4 plus 1/4, which is half the time, which is a 50% chance. Now, let's try parents with different genotypes and look at them using the Punnett square. Let's cross a heterozygote and a homozygote. On the Punnett square, we can see 2 possible genotypes, tall, tall, and short tall. And these occur each half of the time. So what are the chances the offspring will be short? 0, because tall is a dominant gene and there are no homozygous recessive offspring from this cross. What are the chances the offspring is heterozygous for the height gene? Well, that's 50%. Now I want to apply these principles to something that anyone can easily grasp: sex determination in humans. Here's a question that maybe some of you will ask in your lives. What is the probability of a woman planning on 3 children giving birth to 2 girls and 1 boy in that order? And I guess you have to be pretty picky if you're going to want to have children of a certain gender in a certain order, but perhaps some people do. Well, the probability of getting the girl first is, of course, 1/2. Getting the second child to be a girl is also 1/2. And then getting the third child to be a boy is also 1/2. So the probability equals 1/2 times 1/2 times 1/2. So that is 1/8. So 1/8 of the time, a woman will have 2 girls and 1 boy in that order if she's planning on having 3 children. What is the probability of a woman planning on 3 children giving birth to 2 girls and 1 boy? And this is not in any particular order. The probability that we determined previously was 1/2 times 1/2 times 1/2 equals 1/8. We don't care about the order. We can imagine 3 different birth orders: 2 girls, then a boy, girl, boy, girl, boy, girl, girl. So in this case, you multiply the probability of each birth order happening times 3, and you get 1/8 times 3, and then the answer is 3 out of 8. That's the probability. So if you're more flexible about birth order, you have a higher chance of getting what you desire. Here I'm showing Thando Hopa, a model and prosecutor who also happens to be albino. Albinism occurs in about 1 in 17,000 people, and it is an autosomal recessive mutation. What is the probability of heterozygous parents having a child with albinism? And if we look at the Punnett square for an autosomal recessive trait, we see that there will be a 75% chance of getting a pigmented child and a 25% chance of getting an albino child. So the probability is 1 out of 4. What is the probability of these heterozygous parents having 3 pigmented children? The genotype of the first child could be either pigmented or pigmented albino. So you add those probabilities and you get 3/4. So 75% chance of getting a pigmented child during your first try to having a child. For the second child, it's the same. And for the third child, it's the same. So you multiply these probabilities and you end up with 27 over 64, which translates into 42% chance. So you have a 42% chance of having 3 pigmented children if you and your partner are both heterozygous for the albinism gene. Now, here again, I'm encouraging you to take a break, relax, get a glass of water, walk around a bit, and then we are going to go on to the second part of the talk, where we will talk about Mendel's principle of independent assortment and how that relates to dihybrid crosses, and do a little statistics. So come on back, pause the video, and then we'll get started. Mendel's principle of independent assortment states that alleles at different loci separate independently of one another when gametes are formed. This is due to independent separation of homologous pairs of chromosomes during meiosis. The take-home message is alleles at different loci separate independently. So how can we look at this and prove this for ourselves? Well, we can look at the segregation of alleles from two different genes, and we can return to Mendel's P's. So we could look at seed shape, which has two different alleles, round and wrinkled, and seed color, which has two different alleles, yellow and green. When we do a dihybrid cross of these traits, let's say we cross in the parental generation a homozygous dominant round yellow parent with a homozygous recessive wrinkled green parent, we would get two types of gametes, the dominant gametes and the recessive gametes. This would lead the F1 generation to be heterozygous. So if we allow those to self, we get four different types of gametes, each coming from the respective parent. So in the F2 generation, the Punnett square looks like this. Here are the maternal gametes, here are the paternal gametes. You add the maternal and the paternal gametes, and you get these genotypes, which leads to these phenotypes, where you would get 9 round and yellow seeded plants, 3 round and green seeded plants, 3 wrinkled and yellow seeded plants, and 1 green and wrinkled seeded plant. So out of the 16 genotypes that you can get out of this, there is a 9:3:3:1 phenotypic ratio. Let's look more closely at the F2 generation from a dihybrid cross. So there are 16 choices, which are depicted here. So if you wanted to know how many genotypes look yellow and round, you would look for these genotypes: round, round, yellow, yellow alleles, round, wrinkled, yellow, green genotypes, round, round, yellow, green genotypes, round, wrinkled, yellow, yellow genotypes. So what is their distribution? So we have 1 out of the 16 carries both of the dominant alleles. 1/4 are heterozygous for both alleles. 1/8 contain 2 copies of the dominant round allele and are yellow and green. Another 1/8 contained are heterozygous for the round wrinkle allele and contain 2 copies of the yellow allele. So overall, 9 out of 16 are phenotypically yellow and round, which is 56.25%. Let's apply the multiplication rule to dihybrid crosses. So let's say you have a cross of 2 pea plants that are heterozygous for the height gene and 2 pea plants that are heterozygous for the seed shape gene. For both of them, you see the 3:1 dominant to recessive phenotypic ratio and the 1:2:1 genotypic ratio. 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive ratio. So what are the chances that offspring will be short and wrinkled? 1/4 times 1/4 is 1 out of 16. Therefore, the probability of 2 or more independent events occurring together is calculated by multiplying each of their probabilities. So let's get into some experimental observations. Let's say you have another pea plant. This is a different species, but it has some of the same alleles. So you can get wrinkled seed coats or round seed coats. And you do a cross where you cross a heterozygous parent with a homozygous parent. The expected ratio of round to wrinkled would be 1:1, right? So you looked at 40 progeny and you found 22 were round and 18 were wrinkled. That's pretty good. The observed ratio is 1.2 round to 1 wrinkled. The deviation from the expected ratio could be due to chance, since it's close, very close to 1:1. But what about in this species, you get 25 round and 15 wrinkled? So that observed ratio would be 1.6 round to 1 wrinkled. Something else is happening here. So how would you evaluate the role of chance here? You could use statistics, in particular, the goodness of fit chi-square test. So what can the chi-square test do? It indicates the probability that the difference between the observed and expected value is due to chance. It does this by calculating the deviation between the expected and observed ratios. But this test cannot do everything. It cannot tell us if the genetic cross has been correctly carried out, if the results are correct, if we have chosen the correct explanation for the results. So what do you need to do the chi-square test? You need to do the experiment, generate the data. The larger the data set, the better. Determine the expected results. Apply the test to the number of progeny. Determine the probability associated with the calculated value. And compare it to the theoretical values that have the same degree of freedom. And I'll discuss what degree of freedom means in a moment. Here is the formula. Sigma means the sum. The formula is the sum of all the squared differences between observed and expected divided by the expected values. DF, degree of freedom, represents the number of different expected phenotypes free to vary in the cross. So these are the, depending on the trait, you can have multiple traits that you're looking at. So here's an example using a trait that shows complete dominance. We have, in the parental generation, a purple plant and a white flowered plant. The purple plant is homozygous dominant for the allele, and the white plant is homozygous recessive. We cross these, and in the F1s, we get two heterozygotes, big P and little p, for all progeny, and they're all purple. Then we intercross these purple flowers in the F2 generation, and we should get 3 purple for every 1 white flower. And the actual data we got is 105 purple plants and 45 white plants. So how does what we observed compare to what is expected? So we expected 112 approximately purple flowers and 37 or so white flowers. So our observed and expected do have some deviation. But is this deviation significant? Is this something that could be explained purely by chance? So we do the chi-square test, and we see that you subtract the observed from the expected for both of the phenotypes, and you square that, and then you divide it by the expected, and you sum these values, and you get 2. So what is the probability associated with this calculated value? We compare the chi-squared value with the theoretical values that have the same degree of freedom. DF, degree of freedom, as I said before, represents the number of different expected phenotypes that are free to vary. That's the number n, and then you minus 1 from them. So if you have 2 phenotypes, n equals 2, here we have white and purple, the formula for the degree of freedom is df equals n minus 1. So therefore, in our case, the degrees of freedom equals 1. Now let's compare our chi-squared value to the values here on this table. So our degree of freedom is 1. So you look for the degrees of freedom on the left here. There's all these chi-squared values across the main part of the table. And then the p-values are on the top. And the chi-squared values associated with these p-values on the right, which are highlighted in the red box, are a significant values. When the probability is less than 0.05, then chance is not responsible for the deviation between observed and expected ratios. So what was our value? Was 2, right? So it was between 0.455 and 2.706. That gives us a p-value between 0.1 and 0.5. So this value is not significant. There's no significant difference between the observed and the expected ratio. So what if the p-value is more than 0.1? What does that mean? So in our case, it is based upon our calculations and comparing that to the table. So therefore, the difference is not significant. And there's a high probability that the difference between the observed and expected values is due to chance alone. So we conclude that no significant difference between observed and expected values. So the chi-square test has been very helpful. Let's test another data set with the chi-square test. Let's look at cats. So we have black cats and gray cats. We know that cat color for these cats is associated with an allele called B. Big B is for black cats and it's the dominant trait. Little b is associated with gray cats and it's the recessive trait. If you crossed a cat that was heterozygous with another cat that was heterozygous, we would expect a 3-to-1 ratio. This assumes the dominance of B. And we would expect 3 black cats for every 1 gray cat. So we did the experiment, we counted 50 cats, and what we saw was 30 black and 20 gray cats as a result of this cross. We would expect approximately 37 black cats and 12.5 gray cats. So there's a variance here. Is this difference statistically significant? So we calculate the chi-square again. Here again, chi-squared equals the sum of the observed minus expected squared over expected. We subtract the expected from observed in black cats, and we get 1.5. We repeat the same calculation for gray cats, and we get 4.5. The sum of these is 6. We'll then compare this calculated chi-squared value with the table. Again, degrees of freedom are calculated by the number of phenotypes minus 1. So we have 2 expected phenotypes, black and gray. So n equals 2. 2 minus 1 is 1. So the degree of freedom is 1. Now we can check all of this with the probability table. So our degree of freedom is 1, which is highlighted at the left. The value we got for chi-squared is 6. So that is in the side of the table where the p-values are significant. So this says that there's a less than a 2.5% chance that the deviation of the cats that we saw was due to chance. So how do we interpret this? A heterozygous cross should give us 3 black cats to 1 gray cat. So the null hypothesis would be there's no real difference between the observed and the expected. If it is not a 3-to-1 ratio, we want to know whether that deviation is caused by chance alone or is there some other explanation. Our p-value was less than 0.05. We got a value of 0.025. Therefore, the difference between the observed and the expected was substantial and significant, and cannot be attributed to chance alone. What could explain this difference? Please give it some thought, and perhaps you might have some suggestions or comments or questions during question time in class. So I hope you enjoyed today's lecture. Here's a little preview of the next lecture, where we'll be discussing sex determination. And here are two flies drawn here, a female and a male. And we'll learn a little bit how flies determine their sex, as well as humans, and possibly some other organisms. See you next time.